Thank you, everybody, for joining us here today. I'm really excited to talk about our work with pet dogs and genomics. My name is Eleanor Carlson. I'm an associate professor at the UMass Chan Medical School and at the Broad Institute of MIT and Harvard. And I'm giving a joint talk today with Kate McGuire, who's a DVM PhD postdoctoral fellow in my group. She's going to give a talk, she's going to kind of follow on me and talk a lot about the genomic technology that we've been using. And I'm going to give everybody the kind of prelude to give all the context on this work. And as I said, our work involves people's pet dogs. We're really interested in figuring out how we can work with pet dogs and their owners to accelerate the development of new cancer diagnostics and cancer therapeutics using kind of the big upswing in the capacity of genomic technologies to look at DNA and measure DNA. And on my beautiful slide here, we've got a whole bunch of our participants in the project. So a bit of background here. So you're seeing some videos here of two different animals. On the left, you've got a wolf puppy, and on the right, you have a dog puppy. I think they're both about 5 weeks old in this particular video. And they're giving us a DNA sample. With the wolves, we had them chewing on a chew toy that we then rinsed off. And with the dogs, we've got them gnawing down on a saliva sample. And it's actually really easy and straightforward to get a DNA sample from dogs, and as it turns out, from wolves. I'm not going to talk about wolves today, but it was such a great video, I figured I may as well put it in. So the reason I wanted to give everybody some context on this is that the history of dog domestication is actually pretty relevant to understanding why they're an exceptional model for studying things like cancer. So dogs were domesticated from wolves, we think somewhere between 10,000 and 20,000 years ago. We're not entirely sure exactly when or where or how this happened. It could have happened once. It could have happened more than once. I'm more on the once side, but not totally convinced yet. But anyway, so that happened a long time ago. And then we probably had, tens of thousands of years of just dogs hanging out near humans. I think the most plausible explanation for domestication is that there was a group of wolves somewhere living in proximity or at least in the same area as humans, and for some reason, some group of wolves decided to start using human garbage or human refuse as a resource. And then evolution took over. Basically, the wolves that didn't run away as quickly got more food and had more puppies, so you've basically got selection on flight distance. And that was kind of the start of domestication, the beautiful story of wolves eating our garbage. But anyway, so then you've got it started, and you have about 10 to 20,000 years of just dogs being dogs. And this is still true today. Most dogs on this planet, probably about 80%, are what we call village dogs or free-living dogs. They don't live in people's homes. They just kind of live in proximity to people. They're not anything to do with the breeds most of the time. And they're just, basically living with humans, but not necessarily in our homes. But then probably at some point in time, dogs started showing up that had particular behaviors that were of use to people. And either intentionally or unintentionally, humans started favoring the animals that were useful to them in some way. And we got all of these really cool diversity of behaviors that we now think of as herding dogs and retrieving dogs and livestock guarding dogs and all the different kinds of dogs out there, even lap dogs. But the breeds, which is what people often really think about when they're thinking about dogs, dog breeds, are actually incredibly recent. For the most part, the breeds only date back to Victorian England, so the mid-1800s. And the reason for this is that a dog breed is actually defined as being a genetically isolated population. So they basically took a group of dogs and decided those dogs were going to be the breed. And at that point in time, you don't bring any new genetics in. It's a closed population. You're only allowed to be a golden retriever if both of your parents are golden retrievers. It's a totally closed population. And so there's much less genetic diversity in those populations, and you kind of get a bottleneck at the founding of each one. And so today, because of what they were doing in Victorian England, we have hundreds and hundreds of dog breeds, each of which is a genetically isolated population with its own kind of frequency of variance going on. And obviously a whole lot of morphological diversity as well, as I'm showing in my picture here. But we also have this third population of dogs. What else is it? Third? We've got purebred dogs, we've got the village dogs and the free-living dogs, and then we also have this kind of intermediate population, which are the mutts. And we haven't actually found a really great word for mutts, as it turns out. That's the one we're using, but they use different words in different parts of the world. But this is basically a dog that is a mix of different breeds. And I'm showing you one here. This is Marty McFly in the video. And if anybody there in the audience knows anything about dog breeds, you could kind of have a guess at what you might think that Marty McFly's breed ancestry is. But the fantastic thing with genetics, of course, is that we can sequence Marty's DNA and come up with algorithms to basically look at what Marty's actual genetic ancestry is. And that, of course, is exactly what we did. And it turns out that Marty McFly is about 30% Dalmatian, looks like 5% Boxer, 5% Chihuahua, and then a whole bunch of unknown. And that's pretty classic for mutts. A lot of mutts have a lot of ancestry from a lot of different breeds. And so you'll see these big chunks of ancestry where we can really figure out what's going on. And then you'll see this other or unknown, which is probably either tiny amounts of ancestry from a lot of different breeds, or it could potentially be ancestry from that non-breed village dog population that in the rest of the world is actually very common. I just give you a zoomed in version here. Okay, so we've got both the purebred dogs and the mixed breed dogs. And in the US, it's about a 50-50 split. Half the population is purebred dogs and half the population is what we call mutts or mixed breed dogs, these mixes of ancestry. And the reason this is interesting in the context of diseases like cancer is that when they founded those breeds, they were often selecting, as I said, there was a bottleneck. They only used a small number of dogs to start each breed, and then they selected very strongly, primarily on aesthetic traits, what the dog looked like, what its fur was like, what color it was, how tall it was, all that kind of thing. And by doing that, they created these bottleneck populations without a lot of genetic diversity within them. It's sort of like all the dogs in a breed are kind of cousins of each other. And in doing that, they inadvertently also created populations that tend to have a high risk of particular diseases, including cancer. And so sometimes the numbers aren't all that great for actually figuring this out, but our best estimates is that things like the greyhounds have the racing greyhound population has about a 1 in 4 chance of developing osteosarcoma or bone cancer. The golden retrievers seem to have about a 20% chance of developing a disease called hemangiosarcoma, which is called angiosarcoma in people, and another 10% chance of getting lymphoma. Boxers also tend to get lymphoma, and I've only labeled a few breeds on here. This is not to say that the other breeds in this picture don't have their own disease issues, but these are the ones that I'm picking out here. And because they have such high rates of diseases, high rates of these particular cancers, that gives us an opportunity to study the cancers in a way that we can't in humans. And so the advantages here of studying dogs for cancer is that, first of all, there's a lot of dogs. Our best guess is that there's about 6 million cases of cancer diagnosed in dogs each year. And when a pet dog gets cancer, they often get very sophisticated clinical care. They often get the same drugs, the same chemotherapies, the same therapeutics that they're using in humans are provided in veterinary medicine. They often get biopsies, they can get radiation. It's all the same types of treatments that we're using for human cancer. So in terms of a model system for developing new therapeutics, they're actually already kind of part of a medical system that's quite similar to ours. Dogs are also accepted as being a preclinical model for therapeutic development in humans. It's an interesting aspect of this research. We tend to have two different kind of pushes at the same time, where we're very interested in developing new therapeutics for the dogs themselves. These cancers are very dangerous in dogs, and many of them have extremely high fatality rates, and new therapeutics are urgently needed on the veterinary medicine side of things. And at the same time, figuring out how that work can actually also benefit the pipeline of new therapeutic development in people. And the final thing is that both the high rates of diseases in particular breeds and the unfortunate shorter lifespan of dogs actually is an advantage for developing cancer therapeutics. One of the projects that Kate's going to talk about after me is the work that we're doing with blood biopsies or liquid biopsies, which is basically technology to try and diagnose and diagnose a cancer and actually figure out what's going on with it from a blood sample rather than actually trying to get a tumor biopsy. And one of the questions with that are things like, can we use liquid biopsy in order to diagnose relapse? When a tumor actually comes back again, could we actually diagnose that earlier and more easily through a blood sample than trying to do imaging and everything? And in humans, to run such a trial, you would have to get a bunch of people that had cancer and then monitor them for many, many, many years to see whether you could actually pick up that relapse. But in dogs, that many, many, many years is going to be a much shorter time span, so you can actually do the studies much more quickly. Similarly, if you were trying to do a study to figure out whether the blood biopsies could detect the first signs of cancer, doing that study in a breed like the Golden Retrievers, where there's a very high risk of those dogs getting cancer, means you can set up a study where you just test every dog every year or every 6 months and basically see when they're going to start, when you can pick up the cancer. And because you know that some percentage of them, 25%, 50%, might end up getting, are likely to end up getting cancer, you're going to have a very large population within that study that actually is useful for answering that question. But there's also some disadvantages right now in terms of cancer research in dogs, and this is what we've been trying to address with our project. The biggest one is just that the studies are too small. Cancer, as many people in this room are going to know, is a very complicated disease. It involves changes in genes, it involves evolution over time within your body, and in order to understand complex diseases, large study sizes are needed. There's also potentially an environmental component to cancer that we don't particularly understand very well. Second, it can be difficult to get tumor biopsies. This is where the blood biopsies come into it. To get a tumor biopsy, you have to do surgery, which means you have to sedate the dog and get a sample and store it appropriately and ship it to the people doing the study appropriately, and all of this kind of work that ends up being quite time consuming, and it's difficult to get large sample sizes for tumor biopsies. And sometimes people just don't want to put their dogs through that kind of thing. And then finally, it can be difficult to capture the environment and the outcomes. I think the outcomes is probably the biggest challenge, is actually, if you're going to do a study to figure out how effective a new diagnostic is, you want to know whether it actually has an effect on the longer term outcome for those dogs, and that means you have to follow them over time. And so the talk that we're giving today, me and Kate, is basically broken down into two parts. Part one, the one I'm talking about, is what we've actually done to try and get those samples in. And part two, which Kate's going to talk about, is what we're basically hoping we're going to be able to do with them. Oh, I put a picture in here of Kate playing with wolf puppies, and I'm not totally sure it was needed for my talk, but I put it in anyway. So the way we actually got all of this started. So a number of years ago, when I was starting my own research lab at the UMass Chan Medical School, I decided I wanted to keep on working in dogs. I'd been working in dogs for years. I was like, what can I do in dogs that would be really interesting? And we'd been working on a project at that point in time involving compulsive behaviors in dogs, which were thought to be a model potentially for OCD in humans. And we'd had some success finding genes that might be involved in compulsive disorder in dogs using a few breeds like the Doberman Pinschers. But we were kind of stuck because we just couldn't get the sample sizes, once again, that we needed. I'm a computational biologist. At the end of the day, I'm all about getting more data in. And we just couldn't get that many dogs that had actual kind of phenotypes, that had diagnoses that we could use in our studies. And we were stuck. We just couldn't figure out how to move that project forward. And at the same time, I kept on having people come up to me whenever I told them I worked on dogs. The first thing they would do was pull out their phone, show me a picture of their dog, and tell me all about their dog. And I was thinking, this is ridiculous. How can I possibly be in this situation where I can't get enough dogs for my study, yet everybody wants to tell me about their dogs? And so what we ended up doing was basically I wanted to capture that information. I wanted to work with everybody that wanted to tell me about their dogs. And so we set up this project called Darwin's Ark. It's an open data project, and anybody can go and sign up their dog. And we actually genuinely do want to know all about your dog. And it was a little bit of a unique project, I think, at the time, at least as far as the work that I'd been involved in. We'd been really focused on studying diseases in breeds because that was thought to be, and at the time it was, the most powerful way to go about doing gene mapping. But that was also why our sample sizes couldn't get that big, because we were trying to get many, many dogs within a single breed. And so I was kind of thinking about it, and I was like, well, if we're going to shift technologies over to sequencing and sequencing everything, then some of the advantages of working in those breed populations aren't really there anymore. And we have this massive population of mixed breed dogs in this country. And it's much easier to recruit people for your studies if every time they tell you about their dog, you can tell them that their dog is in fact special and important, and you really, really want them in your study. And that was what we could do. We were just like, we are going to take all of the dogs. Any dog can sign up. And once you sign up, I think we've got over 20,000. I need to actually get the numbers on it. We've got it in the 20,000 somewhere of dogs signed up for this at this point in time. So I have this kind of, I used to call it a dirty secret, but I tell everybody all the time, so it's not a secret anymore. But even though I've worked in dog genetics since I was a graduate student, I've never actually owned a dog. And I am, in fact, a cat person, as evidenced by the cat behind me. Anyway, so I have a little fake account on Darwin's Ark so that I can take screenshots of it, and I put my cat in there. My cat was named Darwin, so that's what you're actually looking at here. But you should not add your cat, because we do not yet have a cat project, although I would love to have a cat project one day. And you can go and add your dog to your laboratory and actually start taking our surveys. You can tell us all about your dog, and we in return will tell you what we are actually finding when we analyze your dog's survey data. You also have the option to order a DNA kit. We also have a free kit wait list that waits for us to get grant money in, which is very hard in science sometimes, and so it's not a very fast process. And so we did set up another option where people could pay to sequence their dog, and basically share their data into this open data set that science, anybody can actually access to do research. Oh yeah, I put this slide in because I gave a whole bunch of talks to people that do fancy genetics, and at the end of it, people would always ask me, so can I sequence my dog? And I was like, I just gave you a whole talk where I told you that you could sequence your dog with us. So I decided I need to put in a much more straightforward slide that says, yes, you can definitely sequence your dog with darwinsark.org. It costs $150. We're doing this very cool new technology called low-pass sequencing, and we will share the data with you when we get it back. I just wanted to mention for people in the audience that are genomics people that know an awful lot about patterns of variation in genomes and all that kind of thing, that we have looked at things like homozygosity and linkage disequilibrium in the three populations, the village dogs, the dog breeds, and then in these mixed breed dogs. And the mixed breed dogs basically occupy pretty much what you would expect, is that they have longer regions of homozygosity and longer linkage disequilibrium than the village dogs do, the totally outbred village dogs, but it's still shorter than it is in the dog breeds, which is actually good for us because it means that when we do things like GWAS, we get smaller regions of association out of it. So back just briefly to the technology that we're using. So we're using this thing called low-pass sequencing and imputation. We sequence the whole genome at 1x in each dog, so 1x coverage, and then we impute variants out of it. And this is something we switched to quite early on when we realized how mixed up the mutts were, because we knew that their linkage disequilibrium was going to be fairly long and we wouldn't be able to tag it effectively with the genotyping arrays that were available. But I do have to say that it's worked even better than we would expect. We get tens of millions of variants out for each dog. We're tagging most of the genome, and the accuracy is actually exceptionally good. So just a few fun insights into what these dogs look like. I had a lot of fun with this as somebody that's done genetics forever, so I keep on putting it into my talks. So we did get all dogs to sign up. It meant that we had a lot more success with recruitment, but it also means we have a genetically complex population. And so we did take a look at each of the dogs. We put together a breed calling panel to assess ancestry, and we've got all sorts of different mixes in there. Everything from the purebred dogs like Caramel on the left. Sometimes the way the algorithm works, you'll have a little bit of other breeds get in here, which is why I put this in here as an example. So she comes out as 93% American Cocker Spaniel. She is in fact 100% American Cocker Spaniel. It's just that the algorithm can get a little confused sometimes. You have that little chunk. We have the F1 hybrids here, like Hubble, who's half poodle and half golden retriever. We have dogs like Coconut, who looks like she's probably a mutt, but then mixed with a Dalmatian. So she's half mutt, half Dalmatian. And then Clarence, who's a super mixed up dog. He's 30% American Pit Bull Terrier, but then he has a bunch of other breeds in there as well. And overall, what we found in the mutts was that so I think it was, oh yeah, that's right. So 66% of the mutts had at least 4 breeds contributing more than 5% ancestry to their genome, which I just find fascinating because every time anybody meets a mutt, they try to guess which 2 breeds it's a mix of, and it's like, yes, there's 2 breeds, but there's actually probably a lot more than 2 breeds. And we actually ran a super fun study. You can actually still check it out at muttmix.org, where we put 30 mutts on there because we wanted to know how well people could guess what breeds were in a mutt. So we got videos and photos and a bit of information about 30 of our participant dogs, put up a website and asked people to come and guess what they thought the top 3 breeds were in terms of ancestry for those dogs. And we had somewhere between 13 8,000 and 14,000 guesses for each of those dogs. I think there was about 30,000 participants in total. And what we figured out is that people are slightly better than random chance, but not a whole lot better than random chance. And we have this great set of 6 dogs in here where I looked at how often they guessed American Pit Bull Terrier for each of them, all the way from Jack, who 1% of people guessed American Pit Bull Terrier, all the way up to Esme, where 60% of people guessed American Pit Bull Terrier. And the really cool thing about this is that all of these dogs are actually about 1/4 American Pit Bull Terrier. And you can really see why people are making the decisions that they make when they call breeds. So Jack has longer fur. People tend to call American Pit Bull Terrier more of its short fur. He's got those kind of like narrower face, I think, that kind of thing, versus Esme, where she's kind of got that short fur, short ears, blocky headed kind of thing going on. I've now talked a bunch about the genetics of these dogs, but the real power of this study is that not only do we actually look at the DNA of these dogs, and I remember telling somebody once, I was like, we could set up this whole website, but if all I got at the end of the day was DNA samples from these dogs, that would actually not be particularly helpful in terms of research, because it's the phenotypes that matter, and without the phenotypes you can't actually do very much with the data. And we do get fantastic phenotypes from the surveys that owners fill out. We have about, I actually don't know how many we have right now. It's probably about 20 surveys that are 10 questions each on there right now that ask about all sorts of different things, including whether your dog enjoys life or not. We insert the animal's name into the question. You'll be happy to know that for the most part, dogs do in fact enjoy life. That is a question that is very skewed towards strongly agree. And so in total, I know I told you at the beginning of this was a cancer talk. We don't have a lot of results from the cancer germline stuff yet, so I'm going to talk a little bit about the behavior germline stuff. So we have 18,000 dogs that we have survey data from. This is a paper that we've got coming out, and about 2,000 dogs with genetic data that we've looked at. I will tell you that people really do like talking about their dogs. We have a fantastic survey completion rate, which is very powerful for us, and we're hugely thankful to our participants for that. There's a couple of things that came out of it. First of all is that owners are actually surprisingly good at telling us about their dogs. I think this is definitely true on the behavioral level as well. People spend an awful lot of time watching their dogs and thinking about their dog's behavior, but it also works for physical traits. We have this very straightforward question to assess height, which is basically how tall are they when they stand next to an average-sized adult person. Ankle high, calf high, knee high, thigh high, or hip high. And that we've got a beautiful ability to do a polygenic risk score prediction of a dog's height off of that one question. And we did validate that it was accurate by comparing it both with, we send a whole bunch of owners tape measures and ask them to measure their dog. And then we also measured a smaller set of dogs ourselves at a dog event and confirmed that what people were reporting in terms of the size is actually pretty accurate. And now we can do a GWAS. I forgot to put the numbers on here, but I think this is a GWAS with about 2,000 animals in it of something called brindle coat color. And I do have to say that we probably kind of overkilled the power on this particular one. So that is what the low-pass sequencing data looks like in a GWAS. We have this incredible density of markers, and this is what it looks like when you have a single gene Mendelian trait that you're doing a GWAS on. But of course, the single gene Mendelian traits are not actually particularly what we're interested in. What we're interested in is complex traits, and we're studying cancer from multiple different directions. And one thing we're really interested in the dogs is why do some breeds get cancer so much more than other breeds? Because that suggests that there are inherited germline risk factors that are actually responsible for that. This is just an adorable picture of a wolf puppy howling, because I'm now not going to give you results for osteosarcoma because we don't have them yet. So instead I'm going to use howling, because howling, much like osteosarcoma or hemangiosarcoma, actually, while it occurs in all dogs, we see a breed difference for this one. We see that some breeds have tendency to howl much less than randomly sampled dogs from the population, and some breeds tend to howl a lot more than randomly sampled dogs from the population. And the one I'm showing you here are the beagles. And actually, if we look in the mutts, we can do a really fun analysis. I'm showing you first here the results for size and then also the results for howling, where we can actually look at how much basically whether a dog that has more beagle ancestry is more likely to howl than a dog that has less beagle ancestry. And for something like size, you can see the small dogs falling out on the left and the big dogs falling out on the right. And for something like howling, we can actually pick out, even with the about 1,000 mutts that we have right now, that bias in the bloodhounds and the beagles that shows that that does seem to be inherited along with that ancestry. We did a GWAS of dog size. We got some nice peaks off of it. I would love to have twice as many dogs in this and see what we find then. But the peaks do make sense. In particular, the one on the right here, which actually is a really nice peak. It's about p-value of about 10 to the minus 10, and it actually sits quite near a gene, SCN3A, which has been linked to oral motor development in humans. And that would actually make sense in the context of howling. So I'm just going to end here by talking a little bit about their work on cancer and to lead into Kate's talk. And so, as I said, the struggle with cancer is that we would actually like to understand not just the heritable risk factors, but also what's going on in the cancer itself. Cancer is a disease of the genome, and it's also shaped both by inherited risk factors, environmental stuff, and probably just bad luck. And so we wanted to be able to collect information from dog owners about their dogs, and not just their germline DNA, but also their medical history, their environment, and also what's actually, when they have a cancer, can we actually find out what the tumor looks like on a genomic level, and potentially one day how it's changing over time. And so we've launched this brand new project called the Darwin's Ark Cancer Project. And this is a bit of a new thing for us. We're actually asking people to sign up their dogs if they have cancer or they've had cancer in the past. And that's a lot more serious than asking whether their dog howls or not. And so we've been trying to figure out how to do that well. And so we're asking whether dogs are diagnosed with cancer, and we're actually, rather than directly just trying to get medical records, ideally we will at some point in time, but we're going to start by actually finding out how much owners can tell us about their dog's cancer. Our experience so far with our Darwin's Ark project is that we have a tendency to underestimate how much the dog owners actually do know about their dog's behavior, and I'm thinking also their dog's health. And so we're going to try just asking them about their dog's cancer and see whether we can actually get some really good information from them that way. And so we ask about where the cancers happened. We ask whether there's been multiple cancers, because in high-risk breeds that often happens. And also we ask basically, whether they're currently diagnosed, when they were diagnosed, what kinds of tests were used to diagnose that cancer. And we do collect our veterinarian's information. And then we also collect treatment information. And it took us a very long time to figure out how to actually ask about something as complicated as this through surveys. And it's going to be interesting to see how well it works. So if you're interested, you should check out the Darwin's Ark Cancer Project, and there's all sorts of more information on that. And we're doing this in partnership with an organization called the IAABC Foundation, which is an organization really focused on animal behavior and animal well-being. And so because this involves often people that have sick dogs, and that's a really difficult thing, we wanted to make sure that we had that kind of component of support as well. We have environmental surveys on there, 3 of them. This was something that we've developed. There's a DVM PhD in my group, Michelle White, who developed these surveys, and we're basically trying to get at information like where the dog lives. The interesting with dogs and environment is that unlike humans, dogs often have lived in one location for their whole lives, and so we can actually connect where they were living when they were younger with the cancer type that they got when they're older, potentially. Features of that home when they were built, et cetera, whether pesticides are used, tick preventatives, and then also food and water sources. We're about to do this really interesting collaboration with Kim Anderson and Peter Hoffman at Oregon State, where we're going to send some of our participants these what we call passive environmental sampling, these silicon tags, where basically you send them this tag, they put it on their animal, the animal wears it for a week or 2 weeks, they send it back, and then we use mass spec to actually kind of quantitatively measure what types of chemicals they've been exposed to in their environment. So we actually get quantitative data on environment. And then finally, and this is where our work with Twist really comes into it, is we're doing this blood biopsy technology. And so we're actually going to trial this thing where we send owners a kit that has a special tube in it to collect the blood biopsy, because you need this special tube to make sure that the cells don't lyse, because the blood biopsy is done with the cell-free DNA. And we're going to send them this kit. We're going to ask them to take the kit to their veterinarian. The veterinarian would do the blood draw and then send the sample back to us. And we're going to use that to actually sequence the tumor DNA and sequence the tumor's exome out of that blood sample, which would actually allow us to not just say this dog has cancer, but actually know what kinds of mutations are in that dog's cancer. And ideally, one day be able to connect up that tumor profile to the germline profile, germline inherited DNA profile, to the dog's environment, and then also to the kind of dog's life history. And I'm really looking forward to actually being able to put all those pieces together. I think it's a really unique opportunity. I should mention we're also collaborating with a project called the Dog Aging Project, which is funded by the National Institutes of Aging to look at longevity in dogs. And they're a separate website, but we're collaborating very closely on figuring out how to answer these questions really well and with statistical power. And so, as I said, these are the blood biopsies I already talked about, and I am now going to just hand this off to Kate McGuire, she's going to pick it up from here and actually talk about that blood biopsy technology and how it works and what we're doing with it. So thank you very much.
Thanks, Eleanor. I'm really excited to talk to you all today about some of the work that we're doing to optimize blood biopsy in pet dogs. And first, I just want to give a little bit of background about blood biopsy. As many of you know, blood biopsy is a really exciting new technique that allows us to sample the tumor genome from a non-invasive blood draw. And in this talk, I'm specifically going to be focusing on cell-free DNA, and that's DNA that's circulating free in the plasma. This happens as part of normal cell turnover. Everyone has some cell-free DNA circulating in their plasma at any given time. But in a cancer patient, some fraction of that cell-free DNA will be derived from the tumor. And that allows us the opportunity to sequence the tumor genome in a very non-invasive way, which opens up a lot of exciting possibilities for diagnostics and patient monitoring. Now, there's been a number of studies over the past few years looking at blood biopsy and its applications in human cancer patients. It's been found that it's very helpful for identifying targetable mutations. And in fact, the FDA has actually approved several companion diagnostics for matching patients to targeted therapies. It's also been shown that blood biopsy can be used longitudinally to monitor for minimal residual disease. And it can actually predict relapse before the cancer is clinically evident on imaging. And finally, it's also been used to identify resistance mutations that are emerging in the context of targeted therapies, which can certainly be useful for making treatment decisions. But despite how promising all of this is, blood biopsy is not yet widely used in the clinic, other than the companion diagnostics that I mentioned earlier. And the reason for that is really that there's a number of limitations, particularly lack of standardization. Basically, there's a lot of things we don't know about how pre-analytical factors might affect the diagnostic quality of a sample. Assays tend to not be standardized, and so we don't really know what the best way to perform those is yet. There's also a lack of sensitivity for early stage disease detection, which makes it more difficult to apply blood biopsy as a screening diagnostic. And there are potential confounders that come up in that setting. For example, the fact that a mutation that arises through clonal hematopoiesis might actually be mistaken for a tumor mutation. And so there are a number of things that we still need to figure out and that we need to optimize. And our group studies dog cancer, and we thought that pet dogs were actually an ideal model for really developing this technology, both for veterinary medicine and for human medicine. And so why are dogs such a good model for cancer? Well, they get many of the same cancers that people do, and they often get many of the same treatments. So they get similar chemotherapy, radiation therapy, even small molecule inhibitors and immunotherapy. But even though they're getting some of the same cancers that we do, the incidence of these cancers is quite different. And so if you look at these pie charts over here on the left, you can see that in humans, carcinomas are by far the most common group of cancers. Whereas in dogs, they do get carcinomas, and over there in the dark blue, but some of the other cancer types, particularly sarcomas, are much more common in dogs. An example of this would be angiosarcoma, which is a very rare cancer in humans, with only about 100 cases a year seen in the US. The analogous canine cancer is called hemangiosarcoma, and that's actually a really common cancer. And we see probably on the order of 100,000 cases per year. And so this difference in incidence of these cancers really opens up the opportunity to study a cancer that might be rare in humans in a much larger patient population by studying it in dogs. Now, dogs also share our environment, so they are exposed to many of the same things that their human owners are. And there have been some studies linking environmental exposures, such as tobacco smoke and lawn pesticides, to cancers in dogs. And so dogs are receiving a high degree of clinical care, and they are being treated for similar cancers to their human counterparts with similar treatments. But there is no gold standard therapy that we know is, absolutely the best treatment. And so there's a little bit more flexibility in which treatments the dogs are given, and the dogs tend to have a more rapid clinical course. And so, the clinical trials can actually be performed much more quickly than they can in a cohort of human patients, which allows for the possibility of much more rapid data collection and optimization. So first, we did some pilot studies to check whether or not performing blood biopsy was feasible in dogs. We worked with Victor Adelsteinson at the Gerstner Center for Cancer Diagnostics at the Broad Institute, and we adapted his technique for blood biopsy for use in dogs. This involves taking a blood sample and separating the plasma, and then isolating the cell-free DNA from that plasma. We then perform ultra-low pass whole genome sequencing, which is sequencing that's targeted to about 0.1x coverage, which is not deep enough to call SNVs and indels, but it is deep enough to get a sense of very large-scale copy number alterations. And so we use a tool called iCore CNA, which takes those copy number alterations and their amplitude and infers the most likely tumor fraction in that cell-free DNA sample. And then for samples that have a high enough tumor fraction, we can potentially move on to deeper whole exome sequencing to call some of those simple somatic variants, which is I'll come back to in a few minutes. So for our pilot study, we had plasma samples from 27 dogs. 6 of these were prospectively collected, and 21 of them were banked. And we found that we were able to isolate cell-free DNA from all of the dogs in this study. Here I'm showing you an example of the iCore CNA output for a dog that had hemangiosarcoma. You have the copy number, the log2 copy number ratio on the y-axis, and then all of the chromosomes along the x-axis. Each dot is a bin, 1 megabase bin across the genome. And the dots that you see clustered around 0 that are blue have a neutral copy number state, and the dots in red are copy number gains, while the green shows copy number losses. And I've also just annotated a few of the important cancer genes, as to where they are in the dog genome. And then this is just another example showing a lymphoma. And again, you can see some of these very large copy number gains and losses. And so having shown that this approach is feasible in dogs, we next really wanted to know whether or not the somatic variants that we see in that cell-free DNA are concordant with those that we're picking up in the tumor. And so for this study, we looked at a cohort of 24 dogs with osteosarcoma where whole genome sequencing had already been performed by our collaborator, Heather Gardner. And we had paired plasma samples from each of these 24 dogs. So we performed ultra-low pass whole genome sequencing on those. And then we ran the tumor in normal and the ultra-low pass sequencing of the plasma with the normal through iCore CNA to compare the large-scale copy number changes. And so here's an example of one of the pairs, the plasma sample on the top and the tumor on the bottom. And what I hope you can appreciate by eye even is just that the many of the copy number changes that we're seeing in the tumor are in fact conserved in the cell-free DNA. And to quantify this, we actually did a Spearman correlation of the log2 read depth ratios between the plasma and the tumor, limiting to the cohort where the plasma samples had a tumor fraction greater than or equal to 10%. And we found that we had a row coefficient of 0.74, so fairly good concordance between the tumor and the cell-free DNA. And that was on par with what the Adelsteinson lab had seen in a cohort of human cancer patients. But we also really wanted to dig a little bit deeper and be able to compare the cell-free DNA simple somatic mutations with those in the tumor. And for that, we needed to sequence deeper. And so that brings us to the canine exome capture. We designed a new capture with Twist, specifically aimed at enabling comparative oncology studies. And as I mentioned, this, for our studies, is really going to enable us to look at the concordance of the simple somatic variants between the circulating tumor DNA and the tumor itself, to really be able to get a sense of whether the blood biopsy could be a proxy for tumor biopsy, and can enable us to actually identify resistance mutations that are emerging as we longitudinally monitor patients. So to design this exome panel, we first started with all of the protein coding exons in the Ensembl canine genome annotation. And then we took a look at the regions that were targeted in the Broad's human exome capture. And if there were genes that were targeted that did not have a one-to-one ortholog that we had already included on the dog genome side, we then lifted those over from the human genome to CanFam3. We also then added some of the targeted regions of interest that had been added on the human side for cancer studies, and that included some of the gene promoters and UTRs for genes like TB53 and other commonly mutated regions from human cancer studies. The final design ended up being 36.6 megabases and had a probe footprint of 40.5 megabases. So we tested the capture. We did a pilot study looking at 12 cell-free DNA libraries, each that had a tumor fraction above 10%. The Broad Genomics platform performed the exome capture, and the samples were sequenced to a target depth of 150x. Overall, we found that the performance was really excellent. Here I'm showing you the target coverage, and you can see on the histogram on the right that the mean coverage was actually 236x, so we got very good coverage. If you see that there is a small, like a tail at zero coverage that we were sort of curious about. There are some targets there that did not get any coverage. When we investigated that further, it turns out that those are mostly targets that don't align uniquely in the CanFam3 genome. But we did find that many of those targets do align uniquely in the CanFam4 genome, and so will be rescued in future versions of the genome. And overall, we found that the metrics were very similar to what Twist had seen with their human exome capture. So we were very encouraged by that. And we really wanted to then address this question of concordance between the tumor and the cell-free DNA. So 6 of the samples that we sequenced were plasma samples that were paired to those osteosarcoma tumor normal whole genomes that I talked about earlier. Um, Basically, we subset the tumor normal whole genome data down to just the exome capture targeted regions, and then we ran variant calling on both the tumor and normal pair, and then the cell-free DNA and normal pair. We used 3 different callers, MUTEC2, Strelka2, and Octopus. And for a variant to pass, it had to be seen in at least 2 of the callers. And so what you can see in this table on the right is, we have the number of tumor variants per sample, the number of cell-free DNA variants, and then their overlap, and the percent of the tumor variants that were seen in the cell-free DNA. And what I hope you can appreciate is in 4 of the samples, we have very high concordance between the tumor and the cell-free DNA, anywhere from 75 up to almost 90% concordance. And then we have 2 samples where we have much lower concordance, and actually no concordance in one case. And this is just another way to visualize that with these Venn diagrams. And what I hope you can appreciate here is that we are calling quite a few more variants in the cell-free DNA than we are in the tumor samples, just in general. And we're still investigating this, but we think it may be consistent with studies that have shown that the cell-free DNA is actually more sensitive for picking up variants that are present in all of the tumor in a patient's body, rather than limiting to just a spatially isolated tumor biopsy sample. We're investigating the 2 samples that have very low concordance. Sample 5 here does have a pretty low tumor content in the tumor sample itself, and you can see that we weren't able to call very many variants there. And so we think that that's mainly what's driving that low concordance. In sample 6, we're still investigating why we have such low concordance, including whether or not we could possibly have a sample swap. So, having seen that the somatic variants actually are fairly well conserved between the cell-free DNA and the tumor, we next wanted to move on to optimizing blood biopsy and seeing whether any of these pre-analytical variables that are hard to address in human patients could be addressed in our dog patients. So we looked at blood draw site and time of day. In both of these pilot studies, we enrolled 10 dogs with various cancer types. In the blood draw site study, these dogs had a blood sample drawn from 3 different veins at approximately the same time point, so the jugular, cephalic, and saphenous veins. And then for the time of day cohort, the dogs had three blood samples taken 2 to 3.5 hours apart over the course of the same day. These samples were taken in Streck tubes. The plasma was separated right away, and then ultra-low pass whole genome sequencing was performed. So for blood draw site, we saw that within each dog, the cell-free DNA concentration decreased with the distance of the blood draw site from the tumor. And that was significant with a p-value of 0.02. Cohort-wide, the tumor fraction trended to lower with distance of the blood draw site from the tumor, but that was not significant. And here on the right, you can see a plot where each color is a different dog, and we're showing the 3 different blood draw sites and the distance from the tumor on the x-axis with the concentration on the y. So you can get a sense of that downward trend. We did not see a significant trend by time of day, so that sort of argues against there being an effect of circadian rhythms or particular timing that would be optimal for sampling. And finally, we were really excited to optimize the clinical applications of blood biopsy in dogs. So we really wanted to see if we could use this technology to longitudinally monitor patients. So for this, we had a cohort of 10 dogs with lymphoma that were treated with doxorubicin and a novel agent. And we had longitudinal plasma samples taken at baseline, so day 0, at day 7 for remission, and then when the dog had relapsed, or their latest visit if the dog was still in remission. And we found that overall, the cell-free DNA yields and tumor fractions followed really well with the clinical course of the dog. This is an example of one of the dogs in the study. So the baseline sample at the top, then the remission sample, and then the relapse sample. And you can see that we have this copy number gain on chromosome 13 at baseline with a fairly high tumor fraction of 72%. That actually disappears completely when the dog's in remission and the tumor fraction drops down to 0. And then in the relapse sample, you can see that copy number gain is actually back, and the tumor fraction has started to rise again. So here I'm just plotting these in a different way. You have each of the 3 time points for each dog on the x-axis, and then the tumor fraction of the sample on the y-axis. If the third time point ends with an R, then the dog had relapsed, and if not, they were still in remission. And what I hope you can see here is that in general, most of the dogs followed this pattern of having a higher tumor fraction at baseline, the tumor fraction dropping down to 0 when they were in remission, and then it coming back up if they had relapsed, or staying low if they were still in remission. We did see that there were a few dogs where the tumor fraction remained low the entire time, and one dog where the relapse sample, the tumor fraction had not risen. And so we're still looking into the clinical variables that might be affecting that. But overall, in the majority of cases, we thought that this was a really promising technique to longitudinally monitor these patients. So in conclusion, blood biopsy is feasible in canine cancer patients. Dogs are a really promising model for developing this technology. We found that the somatic variants are highly concordant between the tumor and the cell-free DNA. We also found that blood draw site may affect the cell-free DNA tumor fraction and yield, while the time of day did not seem to make a difference. The cell-free DNA tracks pretty well with the disease burden and response to therapy. And this new canine exome capture was very useful in enabling comparative applications, including, our investigation of cell-free DNA. And so we're excited for the community to be able to use it. And finally, I just want to acknowledge all of the people that worked on this project. It was a big collaborative project. I especially want to thank Eleanor and Cheryl, my mentors, and all of our funding agencies. And with that, I'm sure Eleanor and I are happy to take any questions. Thank you.